Welcome to Unit 5, Section 3, Protein Synthesis, Transcription and Translation. Protein synthesis, the central dogma of biology, transcription and translation. DNA, RNA, protein, trait. Transcription is a process that occurs in the nucleus. In transcription, a single gene from the DNA molecule gets transcribed or copied exactly onto a much smaller mRNA molecule. So the mRNA can take the gene out of the nucleus and to the ribosome. At the ribosome, translation occurs, where that single gene gets translated into a protein, and that protein, having a specific shape, determines the trait. Before we start discussing the details of transcription and translation, we need to understand that DNA is a language, just like English, Spanish, Chinese, or even computer code. Human DNA contains around 3 billion base pairs, or DNA letters. That would be like the alphabet of this language. And these letters make up around 20,000 different genes. Each gene is a small segment of DNA that codes for one protein. In a gene, just like the English letters CAT code for this little guy here, a cat, the DNA letters CAT code for the amino acid valine. In the DNA language, every 3 DNA letters codes for one amino acid. As you can see here, CAT codes for valine. AGG codes for the amino acid serine. CCG codes for the amino acid glycine. And GGA codes for the amino acid proline. 100 to 6,000 amino acids bond together by peptide bonds make up a protein. Imagine, that's 300 to 18,000 DNA letters to code for a protein in each gene. Here we could see the DNA code again. GGT coding for this amino acid, and this one. CCC coding for this one, and you'd have about 100 to 6,000 bonded together by peptide bonds, all to make a protein. DNA, RNA, protein, trait. Now, there are 20 amino acids that make up human proteins. Now, each of these amino acids has their own individual molecular shape. As a result, the type and sequence of these amino acids will determine the shape of the protein. And the shape of the protein is what results in its specific function. Specific shape, specific function. Just like we discussed earlier in Unit 2 regarding enzymes. An enzyme's specific shape fits in a lock and key manner with substrates. And if the shape changes, then it cannot do its job. So the exact sequence is extremely important. But there are 2 problems that need to be solved in order for the DNA code in a gene to produce a protein. Problem number 1. DNA is too big to leave the nucleus. That means that the information to build proteins has no way to reach the ribosomes, the protein factories that are found in the cytoplasm of the cell. Here we see the DNA. The DNA is inside of the nucleus. The DNA is too big to fit through these little nuclear pores. And here is the ribosome. So the first problem is, how do we get the information from a single DNA gene out of the nucleus and to the ribosome, so the ribosome can have the instructions to make a specific protein? DNA is like a giant 600,000 page cookbook that is too large for you to carry out of the library. For perspective, think about carrying 600 of your textbooks. While a gene that codes for one protein is like a single recipe, and the protein is like a single meal. How do you get the recipe outside of the library when the cookbook won't even fit through the door? Or using genetic terms, how do you get the gene outside of the nucleus when the DNA won't even fit through the nuclear membrane? The solution to this problem is called transcription. Transcription is the first process of protein synthesis. Protein synthesis being the central dogma of biology. How DNA gets copied onto an mRNA molecule to produce a protein that results in your traits. DNA, RNA, protein, trait. To transcribe something is to copy exactly. Back before the printing press, there were people called scribes that would copy every single official government document and business document, and even religious documents, letter for letter, word for word. So the word transcription tells us by its very meaning that we're talking about copying the information of a single gene. Letter for letter, and in quotes, word for word, onto a much smaller messenger RNA molecule, or mRNA, so the gene can leave the nucleus. Here we have the solution to our first problem. Now the problem was that DNA was way too big to leave the nucleus. So in order to get a single gene out of the nucleus, we need to copy the gene, letter for letter, and word for word, copying it exactly onto a much smaller mRNA molecule. So the mRNA molecule could leave the nucleus and deliver the message, the gene, to the ribosome. So that the ribosome would have all the instructions that it would need to make the exact protein with an exact shape to fulfill an exact trait. During step 1 of transcription, the enzyme helicase unzips the hydrogen bonds of the DNA molecule. In this picture, we see helicase pictured as a yellow triangle. And here we have the double helix, with the blue line representing the sugar phosphate backbone on both sides of the DNA molecule, and the nitrogenous bases being represented by the blue, yellow, red, and green rectangles. And between the nitrogenous bases, these dots represent the hydrogen bonds. Now, the helicase would come and break these hydrogen bonds, exposing this single strand of DNA, which becomes the DNA template in order to make the mRNA copy of a gene. During step 2 of transcription, the enzyme RNA polymerase attaches to the beginning of the gene, as you can see here in this picture, and uses the template strand of DNA and loose RNA nucleotides to make an mRNA molecule that carries the copy of the gene. In this picture, you can kind of see it happening. You have the DNA template, TAG, GTT, and so forth, here being used, and then the mRNA nucleotides attached to it. A bonding with T, U with A, C with G, C with G, A with T, and so forth. Notice how in the mRNA, instead of T's, we have U's. During step 3 of transcription, the mRNA molecule then leaves the nucleus, carrying the copy of the gene to the ribosome. You can see this happening in this picture. Transcription happening in this little bubble where the helicase unzipped the DNA, the RNA nucleotides came to this area, and RNA polymerase put them together, and out comes the mRNA molecule, carrying a single gene with the instructions for a single protein that has a specific shape to form a specific trait. And this mRNA carries the gene outside of the nucleus, through the nuclear pore, to the cytoplasm, and eventually to the ribosome, where the protein is made. In summary, here we have protein synthesis, transcription, and translation. We just covered transcription. What happened during transcription? DNA, or more specifically, the gene, one gene in the DNA molecule, gets copied onto an mRNA molecule. When does this happen? All of the time, trillions of times per second in your body. Where does this happen? In the nucleus, because the DNA is too big to leave the nucleus. Why does this happen? To send a gene out of the nucleus.